Okay, so I'll just hop right in. I'm going to give a brief overview of the one pipeline that I had kind of mentioned earlier, um, and talk about the data that we have available on Dandy for which resulted from this pipeline, and also talk about some data that we are planning to upload in addition to what we already have. So as we briefly touched on, we are a multi-imaging, multi-site pipeline, and this is done in collaboration with Mass General Hospital, Boston University, um, Mount Sinai in New York, and London, Italy. And so for as just overall goal for this project is to develop a cellular atlas of the human cortex. And kind of the first step in this pipeline is collecting magnetic resonance imaging at 7T. And we typically image a whole hemi control brain with the PA removed and BA44, 45 intact. And this is done at 120 micron resolution. And then the sample is blocked in that BA44, 45 region at 4 by 4 by 2 centimeters. And we collect OCT imaging around 10 micron. And then the sample is shipped to Italy with thick slices of about 450 micron, where they collect light sheet microscopy. And this is done at about 3 micron. And finally, we do stereological counting on the light sheet microscopy data. And just to kind of give a general understanding of why we collect this data and what it's used for, at the MRI level, we use this data to establish a macroscopic reference, coordinate system of the laminar and cytoarchitectural boundaries. And then we do cell counting, which is obtained with traditional immunohistochemistry, as well as a custom-made inverted confocal light sheet microscopy for 3D imaging at the cellular resolution. And then finally, polarization sensitive OCT or optical coherent tomography then enables registration of distorted histological cell typing, which is obtained with light sheet microscopy and then can be registered back to the MRI coordinate system. And so to go a little bit in depth of each imaging modality, just briefly, for ex vivo MRI, the acquisition is about 65 to 75 hour scan sessions, where we acquire 5 clip angles, B0 field maps, a series of changing voltage scans for the B1 transmit maps. And then these field maps are used in the post-processing pipeline for MRI, where we correct for magnetic and radio frequency field inhomogeneities. And these distorted images, these distortion corrected images, sorry, not only improve the SNR for high quality images, but they also help to accurately identify and delineate the layers, the cortical layers and vessels in gray and white matter. And optical coherence tomography, for people who aren't familiar with this imaging modality, it's a technique that uses low coherence light to capture micrometer resolution and cross-sectional images of tissue. And this is performed on a dissected block that I have mentioned, which includes Broca's area to distinguish the cortical layers and cytoarchitectural boundaries at the mesoscale level. And this tissue is imaged and then cut, and this helps us to preserve the original geometry. For light sheet fluorescence microscopy, this is a fluorescence microscopy technique with high resolution optical sectioning. And this uses a thin sheet of light to excite fluorophores in the focal volume. And in our case, this imaging modality acquires the 3D anatomical maps of cell types based on immunostaining identification. And we also refer to this data and we note it on Dandy as SPIM, the selective plane illumination microscopy. And we do manual labeling after acquisition. This helps drive registration. For MRI in particular, we'll label the supra and the infragranular layers of the cortex, as well as the vascular structures. And this has been done in about 12 samples. And for the light sheet fluorescence microscopy, we've labeled 1 sample and we labeled the cortical layers, as well as the vasculature. This was done in about 16 slices for the light sheet. And so when we talk about driving registration, for example, vessel-based registration, we will register the OCT labels to the MRI using a semi-supervised learning technique. And then common features that can be found in the histological and the OCT images allow for a non-linear registration of the histological images back to the OCT and the MRI coordinate system. And so this is just kind of a shout out to the teams. At the Martino Center, we have Bruce Fischel and company. At Lens, we have Francisco Pavone and his team. Boston University includes David Boaz and his team, and Mount Sinai is Patrick Boff and his team. And to get into the data that we have available on Dandy, this is our landing page for all data that we share across sites and modalities, and you can use the Dandyset actions to interact with our data. And so we have raw data and derivative data available on Dandy. When we talk about raw data, this is data that can be manipulated and it's at its kind of rawest form after acquisition. And in our raw data directory, we have a data set description that describes what data is available here and what kind of files you can find. And the way that we've kind of organized these raw data directories or folders they're divided by subject, and then within each subject, we have the imaging modality and the files that correspond to said modality. And so, for example, for MRI, the raw files that we have here are the individual echoes, and these are the distorted images that we initially acquire when we collect our data. And we typically also collect field maps, B0 field maps and changing voltage scans, which we plan to upload to Dandy as well, since these files are used to generate derivative data, which we'll get to here. And for our derivative data available on Dandy, we organize this in a way that describes the processing method, since we have so many different kinds of data and they're all processed in different ways. And within each folder of the derivatives folder, there's a data set description that describes what the name of the method is, what is done with this method, and what kind of files you can find output of the method. And so, for example, we have the EPIC folder for the derivatives, and this is a kind of processing method that we do for the MRI. And within this EPIC folder, we have the files for every given subject that have been processed this way, and the files that are outputted with this. And for MRI in particular, this EPIC method is undistorted B0 and B1 corrected data used with these EPIC scripts. But you can also find, if you're interested in kind of imaging modalities other than MRI, you can find this NBF stereology. We have labels for spin available as well. And we plan to upload the manual labels that we create as well for MRI and OCT. And to dive a little bit more into the MRI derivatives, this EPIC method in particular, we have one sample that has made it through all the way through the pipeline from MRI to stereology, and that is subject I-46. And this is a bit of an older case. So while we collect 120 micron isotropic MRI now, this sample has 150 micron isotropic, but this is a variable flip angle scan, which has been processed through a series of scripts to compute synthetic volumes, volumes, which have been corrected for that B0 and B1 magnetic fields inhomogeneity. And the way that that's done is using those raw echoes, which you can find in the raw data folder on our Dandyset. We undistort those echoes using the EPIC scripts, which we will also upload to the LCM GitHub by inputting the B0 field maps and edge preserving prior. And then these B0 corrected undistorted echoes, as well as the B1 transmit maps, are used to estimate T1, T2 star, and PD with FreeSurfer's MRI MS fit farms. And then this also uses the T1, T2 star, and PD volumes to synthesize images at the acquired flip angles. And fitting and synthesizing has the effect of removing the B1 transmit, which is the inaccurate flip angle, as well as averaging noise across flips and echoes to improve the SNR of our images. And as I had mentioned, if you want to interact with this data and manipulate it in your own way, you can always find our data in this raw directory file, which has each subject listed, and again, each imaging modality that has been collected on each subject. And as I had mentioned, we plan to upload the B0 field maps and the B1 transmit field maps, as well as share these EPIC scripts for correcting the MRI data.
So our data sets, just spin microscopy. Um, the physical pipeline starts out with a brain that's sliced on our own hand designed vibratome, slices it into 2 millimeter slabs. Then the tissues transform using processes to clear it and to convert it into a elastic polymer, binding the proteins to the polymer. And then it gets run on our hand-built light sheet microscope. The light sheet microscope's oriented obliquely at a 45-degree angle to the stage motion in order to give it both in order to give it a stage motion, a long stage motion path for the whole hemisphere slabs. And also because that way it doesn't, the illumination arm doesn't have to illuminate through the whole slab. So it's at this 45-degree angle, which requires a little extra processing to render orthogonal volumes. So you have the camera angle is for the 45-degree angle to the X motion. So you get these image planes and you get this sort of parallel pipette structure, which has to be reorganized into stacks. And then the stacks have to be have to have the artifacts removed. There's a striping artifact that happens when something's upstream of the light sheet path. And this is done using wavelet decomposition to remove the artifacts. And after that, you get these stacks, and they have a little bit of overlapping region, which has to be aligned. There's the rough stage alignment, but usually there's a few microns off because of the stage isn't perfect. So you take patches out of there, and you go through this process of finding the nuclei in the patches and registering those using correlation coefficient. After that, you can take multiple rounds of stain for the tissue. The tissue can be have the stain washed out of it, restain, and we have a multi-round alignment application for that. Again, it finds the nuclei and does a gradient descent to align the nuclei to the from one round to the other. The slab, you have individual slabs, and the slabs don't have any overlap. We use the blood vessels in between each slab, finding the blood vessels in the top and the bottom, and these form these constellations where the nearest blood vessels have angles and distances to them, which form the features which can be matched from one slab to the other. And so those, once you have those, you can have a registration between slabs, and that can be built up into a transform that can be saved to work one slab to meet the other. We store our data using the new BIDS microscopy spec. The individual stacks are stored in NGFF, which has underneath it a ZAR array, which looks very much like a Python NumPy array. And the stacks, according to the bid spec, have a sidecar JSON file, which stores the offset alignments between the stacks, so that you can you can reconstruct the stacks on the fly and for display and to analyze them. Right, and ours, and yeah, so ours are yeah, and our data is stored in Dandy. We have a couple of use cases for it. The first one is NeuralGlancer, which I don't know if you guys are familiar with, but this is a stack of Dandy data, and you can zoom in and out. And if you zoom in, you'll probably see a little bit of blockiness. There's a variety of bitmap levels that it uses to allow you to view the data at its most coarse resolution and zoom in to see it at its finest. So this data is publicly visible, served by a web server, so that anybody can use it and view it. Other use cases that we plan to do are fiber tracing and characterizing different cell populations, and these all can be done hopefully within Dandy on the on cloud architecture. So that's pretty much it. We've have a variety of people, including Satra and Marie Helene, who was instrumental in bids, and Casey is my PI on this. That's it for me.